Hi everyone. So today we'll be starting with organic compounds. So you know that now when we speak about organic compounds, we have carbohydrates, lipids, proteins, nucleic acids in our syllabus, right? So today throughout this video, I'll teach you all the organic compounds, all the information that you need to even write an essay. So mainly we'll focus on making a mind map kind of a thing with this with these organic compounds. So whenever you have to write an essay, you don't have to separately recall the points. You can simply go through this video, and while I do this itself, you can also make your own mind map with me. So at the end of this video, you'll have a mind map for each carbohydrate, proteins, lipids and nucleic acids. And before the exam or before any other unit test or anything, you can soon recall everything that's in the resource without having to go through it each sentence by sentence. So in this way you can remember these things will better, because I'm going to teach you in a way like using mnemonics. That means like shortcuts. And also I'm going to teach you in a way that you can answer any MCQ, because we are going to learn it in a way that you won't forget it any sooner, right? So even for the exam, now in your final exam, you might have one of these organic compounds as an essay. So it's always better to be very thorough with this, because this is a very the introductory bit of biology. So if you are okay with this part in a very thorough way, then you can have that confidence. Okay, I know the introductory part well, so everything in biology is going to be very similar to this. I mean the format. If you know how you can make a mind map. By mind map I mean you write the central topic here and then you're going to make a mind map in a way like a flow chart. We know no one remembers things if you are going to recall things using paragraphs and sentences. That's very rare. But if you make it in this way, in a flow chart kind of way, a mind map, then everyone can recall things better, right? So even during your essay, when you're writing an essay, you'll think, okay, there was something on the right side that I have missed to write in my essay. Or when you repeat this mind map technique for 2 to 3 essays, then you realize that okay, I'm missing something that I wrote usually in my short notes in the down part. So for biology, creating short notes is also somewhat of a waste of time. Why? You have to, every point is important. There's nothing that's less important. So every point is important when it comes to an essay. So if you are going to make a short note for biology, that's also a bit unrealistic. So what I would recommend is make a mind map with all the key points. So when you look at that mind map, you should be able to teach like what I'm doing now. You should be able to teach this to a person who doesn't know the context in a way that they can understand everything from the basics. So that's the whole point of this video, right? So we'll start with carbohydrates. So we'll now start with carbohydrates. Okay, so carbohydrates, now what's the first thing that comes to your mind when I say carbohydrates? Now the very basic stuff that we learned in O levels and grade 9 and all that stuff like floury stuff like bread, noodles, those are stuff that are rich in carbohydrates. Now we are going to learn what happens in that in a chemical basis, what is there in the deep, when you zoom it in, and if you consider carbohydrate molecule, what is there in that. So that's what we are going to learn under carbohydrates. So now when they speak about carbohydrates, you have to, now this is the central point that we have for our mind map. Okay, so now firstly we are going to speak about the composition of it, right? So when we speak about the composition, what's a composition? How it's made of. Composition is how something is made of. So when you speak about the composition of something, you have to say now what is it made of. So here carbohydrates are made up of carbon, hydrogen and oxygen. So these 3 elements are only going to make up carbohydrates. And then they have also included a point about the hydrogen to oxygen ratio. Hydrogen to oxygen ratio. Now, if there are 2 molecules of hydrogen, 1 molecule of oxygen. So what is that similar to? Like in water, right? So now in water, you can see there's 2 hydrogen and 1 oxygen. So 2:1 ratio is there in carbohydrates like in water. You have to mention like in water. Okay, and then you are going to also speak about now this is one point. Okay, this is one point. Then the ratio is another point. Then the third point is about what? I'll put the points in crosses, then it's easy to understand. Okay. This is one point, this is one point. And the third point when we speak about composition is about the general formula. So what's the general formula? CX H2O Y. Now this is the general formula of carbohydrates. And now these three things only come under composition. That means now when you speak about carbohydrates, if someone asks what it's made up of, these three things are what comes to your mind. Okay, and also there's a very important point. Now in the resource that's given as the very first point, what is it? That's the most abundant organic group. So most abundant organic group on earth. Okay, now the most abundant what? The abundant organic compound. Okay, organic compound is the most abundant organic compound. Remember, not most abundant element. Now in your MCQs and all that, just to confuse you, they can say the most abundant element is carbohydrates. Now you might think, okay, this is something very similar to what's in the resource, and you'll put it right. But no, remember this, organic compound which is most abundant is what? Carbohydrates. Okay, the terms are very important in bio. Even if you write the correct thing, if you don't have the key terms, then you don't get the answer, right? You don't get the mark for it. So organic compound is a key term in that. And then we are going to speak about the three groups of carbohydrates. Now this is also something we learned in O levels, right? O levels also we have this thing. So it's not like everything is new in bio. You're like learning something a bit more advanced than what you did in O levels. Okay, so don't get scared of the content saying this is a lot to remember and all that. It's a bit more than what we have in O levels. Okay, so when we come to the three groups of carbohydrates, first of all we have what? We have monosaccharides. Monosaccharides, I'll write in short form mono. And then we have disaccharides. And then we have polysaccharides. Now what is saccharides? Saccharides means that means sugars. Okay, so what is mono? Mono means one. What is di? Di means two. And what is poly? More than two. More than two means you call it poly. So if there is one sugar, you call it monosaccharide, two sugars disaccharide, and more than two you call it polysaccharide. So that's a basic idea about carbohydrates. Now then you have to speak about the general features of these carbohydrates. So with this itself we can continue that part. So what they are saying here is that mono and disaccharides are going to be what? Sugars. But we don't consider polysaccharides as sugars. Okay, now although the name saccharide is there in this, now this might sound confusing to some of you all, but listen to this properly. Although we have saccharides here, I told you saccharides means something like sugars, but although we have the term saccharides here, polysaccharide is not a sugar. What are sugars here? Only mono and disaccharides. Polysaccharide is like a polymer. Okay, polymer means if you have, now if there's something big that is made up of very small repeating units called monomer. So this whole thing is going to be called what? A polymer. Now this polysaccharide is not a sugar but like a polymer. Okay, so if they ask which of the following are sugars, you don't say polysaccharide is a sugar. You say only mono and disaccharides are sugars. Okay, so these are the basic information that you need to know about carbohydrates. So now see, when you take the topic itself, if you are going to read it directly from the book, can you remember it properly like this? No. Why? Now you have put it into a format, then you remember it better. So we'll quickly go through this. See, carbohydrates, what's the basic information? That we have, it's the most abundant organic compound. Then we have the composition of it. So carbon, hydrogen and oxygen is the elemental composition. And then that elemental composition consists of hydrogen to oxygen. The ratio is 2:1 as in water. So we'll put it here, 2:1 hydrogen to oxygen ratio. Then the third point that we have is the general formula, CxH2O(Y). Okay, and then we have three groups of carbohydrates, like we know this already. In O level also we have it: monosaccharide, disaccharides and polysaccharides. And then I told you mono and di are going to be considered sugars. Polysaccharide is not considered as a sugar. So that's the introductory bit for carbohydrates. Now after this we are going to learn about each type. Now I told you there are three types of carbohydrates, no? Mono, di and poly. So each type we are going to learn each of them in detail. Okay, so this is the introductory bit of carbohydrates. Now we move on to each type. So we have monosaccharides. I'll put mono. Okay, now it's better if you can also make a mind map while I'm teaching this, then it goes better into your head. Okay, so now we move on to monosaccharides. Okay, so I told you what is monosaccharide. Monosaccharide means you have one sugar. Okay, di means 2, poly means more than 2. So now we are going to make a mind map for monosaccharides. So first of all, now we had a similar introduction for carbohydrates. What did the point here be? Most abundant organic compound. For monosaccharide also we have a unique point to remember like that. What is it? That's the simplest form of carbohydrate. Okay, simplest form of carbohydrate. That's what you have to remember about monosaccharide, the first point. Then second one we can speak about. Now for carbohydrates, this side what did we speak about? This side we spoke about the composition, right? So here for monosaccharide also we'll speak about the composition. Then it's like, similar points to, then you can generalize the concept here. So firstly we can say composition. Okay, so composition. Under composition for monosaccharides, now there we spoke about, now here monosaccharide is also a carbohydrate, so that's going to have carbon, hydrogen, oxygen. Now we are going to speak about the number of carbon. Now number of carbon is going to be, carbon number is going to be 3 to 7. That is 1 point. Okay, carbon number is going to be 3 to 7. That is 1 point. Then the second point is the general molecular formula. Now there we had CxH2O(Y). No, here the composition of monosaccharides. Now that previous one that we did here, this one CxH2O(Y), that's a common formula. This is a common formula for all carbohydrates. But now we have a unique formula for monosaccharides only. What is that formula? CH2O(N). So this is the formula for monosaccharides. Okay, so that's it about the composition. Then we are going to speak about here for carbohydrates. In the lower part, what did we speak about? We spoke about the three types of carbohydrates. Now here we are speaking about the features of all monosaccharides. So all monosaccharides have some features. All have some features. What is it? What are the features? Now this was also there in O level. What are the features that we can remember? Now if you just think about sugar, normal sugar that you have, that you add to tea. Now sugar cubes. Okay, so sugar cubes, what can you say about those sugar cubes? You can say they are crystalline, right? So they are solid. No, in the basic form they are solid, so they are crystalline. Firstly, they are crystalline. And then after crystalline, you can say they are water soluble. Why? You are going to add the sugar to water and mix it, and that's how you make tea. No, why? Because sugar is going to dissolve in water, so they are water soluble. And also you can say something called reducing sugars. Reducing sugars. Okay, there is this term. This might sound new to you. Reducing. Just remember it. Monosaccharides are going to be reducing sugars. Now I told you mono and di are only going to be mono and disaccharides are only going to be sugars. Out of that, all monosaccharides are reducing sugars. Remember that. Now, if they ask for the features of all monosaccharides, what do you have to recall very quickly? Just the sugar that you add to your tea. Why? It's going to be solid. And then you are going to add it to the tea. So how can you add it to the tea? By dissolving it in the water. So it's water soluble. And also you can say it's reducing sugars. Okay. Now this is about the features common to all monosaccharides. Now after this we are going to speak about how you can classify monosaccharides. Okay. You're going to classify monosaccharides further. So now I think it's better if we put this part here. Why? For carbohydrates also we classified something down. No, I mean the format. Okay, so features are going to come this side, and here monosaccharides, you're going to classify monosaccharides into two types. Okay, firstly you're going to classify monosaccharides. What is classify? Classify means categorizing. So you're going to classify monosaccharides into groups based on what? Their carbon number, like the number of carbon number. Based on the carbon number, you're going to classify monosaccharides. So I told you how many carbons can be there in monosaccharides. I told you here 3 to 7. So now for each carbon number you are going to give a common name. Okay, like a group name you are going to give. If the monosaccharide has 3 carbon, then you have another name for it. If it has 4 carbon, you have another name. If you have 5, you have another name. If you have 6, you have another name. Remember, not only 6, we have 7 maximum carbons in a monosaccharide. But we don't have the name for the 7th carbon. Okay, so only till the 6th one we have in our book. Okay, you don't have to know anything that's beyond our resource. So firstly we'll speak about 3 carbon. Now this is like, you know, now you can like understand 3 carbon. 3 means what? Tri means 3. So os, the term os is going to come, os, with that prefix. Okay, so triose means what? 3 carbon. So triose means 3 carbon. Now for triose you have to give examples. Okay, so we have glyceraldehyde. Now these names might seem a bit complex for you. Why? You're learning this for the first time, no? But after you start doing chemistry, like, you know, towards the end of grade 12, then these names are going to be very normal for you. For now it will be a bit complex, so write it like 2 to 3 times and you'll remember it. Okay, and another example is going to be phosphoglyceraldehyde. There's a phosphor part added to this glyceraldehyde. Okay, now this phosphoglyceraldehyde is a derivative of glyceraldehyde. What's the meaning of derivative? Derivative means now there's a normal thing. Okay, there's an original thing. If you modify it a bit and you make a new thing out of it, now there's a small box, you color that box and now it's a new box. Okay, so what I'm saying is, this is an original thing, you modify it a bit and you make a new thing out of it. So this is a what? Derivative. Okay, something that is obtained from the original is called what? Derivative. So here phosphoglyceraldehyde is a derivative of what? Triose. Now what is it actually? Now see, when you add phosphor in front of glyceraldehyde, you make phosphoglyceraldehyde. Now what is glyceraldehyde? Glyceraldehyde is a triose. Therefore we say that phosphoglyceraldehyde is a derivative of what? Triose. Okay, so these are the points you have to remember under 3 carbon. 3 carbon is triose, triose examples we have glyceraldehyde and phosphoglyceraldehyde. Phosphoglyceraldehyde is a derivative of triose. Okay. Now we are going to move on to 4-carbon. So 4-carbon, you call it tetrose. Okay. So tetrose means 4-carbon. Now for tetrose you have an example. What's the example for tetrose? You have erythrose. Erythrose. This ose means, it means sugar, something like sugar, okay? Glucose, lactose, cellulose, tetrose, erythrose, okay? Ose means like sugar. So erythrose is an example for tetrose, okay? Now this triose, tetrose, that's like a common category name, like vegetables, fruits, something like that. These are examples for that, okay? So I'll box the common category name, then it's easier to understand, okay? Tetrose, you have example erythrose. Now they have given a point about erythrose. What is it? It's rare in nature. So just remember something like this, okay? Why have we put this remark here? To show that, okay, this is rare in nature. This one is special. Why? Because it's rare in nature. It's a point. So in your MCQs they can say, for example, I'm telling you how they can question this. They can be like, which of the following monosaccharides is rare in nature? And they can give phosphoglyceraldehyde, erythrose, deoxyribose, ribose. Then you have to remember, 4-carbon only we put this sign, which means there's something special about it. What was the special point you have to remember using that? It's rare in nature, okay? Now this mind map, you don't have to write everything that you know about each point, okay? These are small triggers. Now see, this sign is enough for me to recall the point that, okay, there was some small point regarding this. Okay, so in your mind map, don't rewrite everything in paragraphs again and again. The whole point of having a mind map is you can look at it and soon recall the points, soon memorize the points, okay? And next time when you read through it, you can remember, okay, there was something about it, something like that, okay? So 4-carbon, we are done with that also. Then we have 5-carbon. 5-carbon, what did we have? How do you call 5? Usually now, if there are 5 angles, sides, how do you call it? A pentagon. So here also pentose. Pent means 5. So pentose is going to be what? 5-carbon. Okay. So pentose means 5-carbon. So pentose also you have 3 examples. What is it? Ribose. And then you have deoxyribose. And then you have ribulose. Okay, these are the 3 that you have to remember. And also now some of you all might have the habit of remembering. Now if there are 3 in a list, just remembering 1. Why? In the exam they're not going to ask all the 3. So if I remember 1, that's enough. No, that's not how you do it for bio. For bio you have to know all the 3 examples. Why? If they give this as an example in the MCQ, you won't know what this is, okay? So don't selectively study when it comes to bio. Remember everything, okay? So ribose, deoxyribose and ribulose. Now like we had for 3-carbon, where we said that phosphoglyceraldehyde is a derivative of triose, here also we have, what is it? Ribulose. Ribulose is going to be called what? Ribulose. And not only 3 examples, we have something called RUBP, okay? RUBP, just remember this short. Ribulose bisphosphate, this is a full term. Now that term you will come across this when you do photosynthesis, not now. So RUBP is a derivative of ribulose. Now this arrow means what? Derivative, okay? Now the same derivative story came here and here, okay? Phosphoglyceraldehyde is a derivative of triose. Similarly, RUBP is a derivative of ribulose, okay? So that's what you have to remember about the derivatives. Then we have the last one. I told you, although we know that the number of carbons in monosaccharide is 3 to 7, you don't have anything about the 7-carbon sugars, okay? Only till 6-carbon. So what's the final one? 6-carbon. 6 carbon is hexose. Okay, 6 hexose. So example for hexose is going to be glucose. Now this is a very common one. All of us know that glucose is a monosaccharide. Why? In O level also we had it. In grade 9 also I think they taught us, right? So glucose, fructose, galactose. Okay. Now these three terms are very common. Why? We all know it. You would have heard this like many times throughout your science units and all that. So this is not something new, but what is the new thing we are learning? We are learning the common name for it, hexose. Hexose means what? 6 carbon. Okay, so that is about the number of carbon and how you categorize them based on the number of carbon. Okay, so we'll quickly go through it. 3 carbon means triose, glyceraldehyde and phosphoglyceraldehyde. Then what do you have to remember? Phosphoglyceraldehyde is a derivative of triose. Now this arrow means derivative. Okay, don't write everything, try to represent it with some small icon or some small arrow or whatever like that. Okay, 4 carbon is tetrose. Erythrose is an example. Erythrose, I have put this shining icon here. Why? To show that it's rare in nature. Then we have 5 carbon, it's pentose. We have 4 examples for that: ribose, deoxyribose, ribulose and RUBP. Okay, and I told you RUBP, we'll learn that later. RUBP is a derivative of ribulose. Then you have 6 carbon, hexose. Hexose, you have 3 examples. What is it? Glucose, fructose and galactose. Okay, so this is about the number of carbons and how you can classify monosaccharide based on the number of carbons. Okay, now I told you there are 2 ways of classifying monosaccharides. Now based on the number of carbons we have done. There's another way, right? So what is another way? Based on the type of carbonyl group. There's something called carbonyl group. Okay, now these are stuff, this carbonyl group, aldose, ketose, these are stuff you learn in organic. You might have heard there's a unit in chemistry called organic. Okay, now you will come across these stuff in organic only. So now it's like something new. So there's no way to teach you all what is carbonyl group, aldose, ketose now, because you all won't understand it now for now. Okay, but like I told you, at the end of grade 12, like towards the starting of grade 13, you'll understand this is something that we learned in bio even before chemistry. Okay, so that's something like that. Now just remember, there is a group. Now in a molecule, if there is a certain group in that molecule, you're going to classify it whether that group is present in that molecule or not. So now some monosaccharides are going to have this carbonyl group. Okay, now there are 2 types of carbonyl group. We have aldo and keto group. Okay, aldo group, keto group. Now don't think it's very complex just because you have 2 different new names coming in. Okay, just remember there's something called carbonyl group, called aldo group and keto group. Now if the monosaccharide has aldo group, you call it as what? Aldose. Okay, now you can understand the pattern. No, O's is going to come everywhere. Okay, if the monosaccharide has aldo group, you call it aldose. If the monosaccharide has keto group, you call it ketose. Okay, now you have to learn examples for aldose and ketose. That's it. So for aldose, you have examples like glucose and galactose. Okay, and also one more thing. Now in the book we have only these 2 as example for aldose. So anywhere else, if you come across any examples outside the book, in some other books or some other tudes, those are not necessary at all. Even if you know those, it's just pointless. Why? No one's going to give you marks for that. So for bio, only what's in the resource. Don't go anything beyond the resource. Okay, I'm just telling this again and again, because when you start doing bio, sometimes some teachers or sometimes in some videos or something, you come across new examples and all that. Even if you know more, just because you know more out of the book doesn't mean that you get more marks. Only what's in the book is necessary. Okay, necessary means that's needed. Don't go to remember examples outside the book at all. There are obviously examples. Why? There's not only, these are not the only example for monosaccharides, no. But what you need is this. That's all. Okay. So for aldose you have glucose and galactose. For ketose you have an example, fructose. So if someone asks whether, now we know other examples, no, ribulose. Whether ribulose is an aldose or a ketose, if someone asks that question, you say, okay, it's not in my syllabus. I don't have to even know it. You don't have to know it. You have to know only whether glucose is an aldose or a ketose, galactose is an aldose or a ketose, and you should know, okay, fructose is a ketose. Okay, so that's all that you have to know when it comes to the carbonyl group and the two types. So now that's about the monosaccharides there. Okay. And also we have now the classification part is over when it comes to monosaccharide. Okay, and we have one more extra point. Okay, I think it's better to put it in under this itself, or we'll put it as a separate point, then that's better. If we put that as a separate point, it will be better. Okay, so there's another extra point to remember under monosaccharide. What is it? Now this is given under the diagram. Now in the resource they have given some big molecules and they seem very complex. Okay, even we don't have to remember. No one has to remember how you draw them and all that. That's just for you to understand, okay, this is how it is. Don't try to like memorize the structure or anything like that. The key point you have to remember from that page, I'm referring to page 10 in the new resource. So in that is that monosaccharides in aqueous media, some monosaccharides are going to be in ring form. Now you know there are two forms, okay, in chemistry and all you'll come across this. There's something called linear form and something called ring form. If the molecule is just one straight line, zigzag line, whatever line, okay, if it is in a straight line or some line form, you call it linear form. And if it's as a ring, that means now that the molecule is there, okay, like this, different groups are there and it's a ring, so that is called ring form. So in aqueous media, some monosaccharides are going to be in ring form. Okay, so ring form you have to remember glucose and fructose. That's all you have to remember from that page. You don't have to remember anything beyond that. In ring form there are two examples. What is it? They have given that with a picture, right? So in solid form they are going to be linear, but linear in the solid form when you add water to it. What is aqueous media? When you add water to it. So when you add water to it, they are going to become ring form. What are the two examples that you have to know? Glucose and fructose. That's it. So we can name this as form. The topic for this subpart is form. Okay. Okay, that's it for monosaccharides. Now monosaccharides, see, what are the stuff you have to remember here? Monosaccharides you have to remember. Now this is all that you have about monosaccharides in your resource. So firstly, monosaccharide is going to be having some key information. Firstly, simplest form of carbohydrate. Secondly, about the composition, what is it made of, how it's made up and all that. So we have carbon number 3 to 7 and also the common general formula is CH2ON. And then about the features that are common to all monosaccharide. What is it? Crystalline, water soluble and reducing sugars. Now after reducing sugars, now how do you remember that these 3 points? Some of you all remember it from O level itself, but some of you all, if you all find it difficult to remember the sugar cube. That sugar cube is solid, no, so they are crystalline. And also you dissolve it in tea, so water soluble, and they are reducing sugars. Then I told you can categorize monosaccharides into two main based on two main criteria. What is it? Based on the carbon number and also on the carbonyl group. Based on the carbon number, you are going to categorize them as, 3 to 7 carbon is the maximum. That's the amount of carbon that can be there in monosaccharide, no. But in our book, only till 6 carbon is given. So 3 carbon is triose. So example, glyceraldehyde and phosphoglyceraldehyde. Phosphoglyceraldehyde is a derivative of triose. 4 carbon is tetrose. Example is erythrose, which is rare in nature. 5-carbon is pentose. Example ribose, deoxyribose, ribulose and RUBP. Now this RUBP is a derivative of ribulose. Then you have 6-carbon hexose. We have 3 examples for it: glucose, fructose and galactose. Now we move on to carbonyl group. Based on the carbonyl group also you can classify monosaccharides, but you don't have to know every example whether it's an aldose or a ketose. You have to know only whether glucose, galactose and fructose is an aldose or a ketose. So I told you there's something called carbonyl group. There are 2 types of carbonyl group. What is it? Aldose and ketose. If aldo group is there, you call it aldose. If keto group is there, you call it ketose, and the examples. And finally, we have another unique point for monosaccharides, the form. So when they are solid form, it's linear in shape. But when you add water to it, it becomes ring form. Okay. So ring form examples are glucose and fructose. Okay. So that's all you have to remember for monosaccharides. Then we move on to the next part, that is disaccharides. Okay. So what did I tell you about disaccharides in the very beginning? I told you disaccharide means it's made up of 2 sugars. If one sugar gets along with another sugar, you call it a disaccharide. Okay, so disaccharide is like a couple. Disaccharide means 2 sugars. Okay, so I'll write the whole name. Disaccharide. Now disaccharide also we have to do a small mind map like we did for monosaccharide. Okay, so what is disaccharide? When 2 sugars are going to join by a glycosidic bond. 2 sugars means what? When 2 monosaccharides are going to join by a glycosidic bond. Okay, now for disaccharide, firstly you have to say that how disaccharide is formed. How is it formed? That's the first part you have to explain. So how is it formed? When 2 monosaccharides are going to join through a glycosidic bond. Now instead of writing 2 mono like this, I'll write 1 monosaccharide is going to join with another monosaccharide, and they are going to form what? A disaccharide. Now how is that? The bond between them. Now there's a monosaccharide here like this, and there's another monosaccharide here. There's a bond between them. What is that bond called? That's called the glycosidic bond. Okay, these are new terms, so don't feel overwhelmed about it. Just remember there is a bond between these 2 monosaccharides, and you call that a glycosidic bond. Okay, for now that's enough. Now you have to know what is this glycosidic bond and how it's formed and all that. That's the next thing you have to know. So that's another topic for us to properly elaborate on. This glycosidic bond, how it is formed. Now we'll go with an example. Okay, now we know this glycosidic bond means, now for example we'll say now there's a glucose molecule. Okay, now I'll just draw a glucose molecule, just an example. Okay, this is not how a glucose molecule looks. I'm just saying example. Glucose molecule has OH, these are going to be there. Okay, so now when this glucose molecule is going to bind with another glucose molecule, okay, this molecule is also going to have like that, similar. Okay, I'm saying in their whole form they are going to have groups like this. Okay, so what happens here is, now this is one monosaccharide molecule, no? This is another monosaccharide molecule. Now in this monosaccharide, the OH group, okay, is going to remove a hydrogen from the other one. Now what is this? 2 hydrogens and 1 oxygen, that is water. So this water is going to be removed as a product. Okay, water is going to be removed, and then you are going to form the disaccharide. So can you understand? The OH group of one monosaccharide and the hydrogen of another monosaccharide is going to be removed as what? Water, and as a result we have the disaccharide. Now if I draw this molecule again for you, This is not the actual molecule, okay? Just for you to understand, I'm drawing it like this. Because if I'm going to draw the ones that are given in the resource, you're not going to understand it. Why? It's very complex looking, okay? So now the same thing is going to repeat, okay? The same structure. Now I'll just draw it again. OH. H is there. Now this OH is gone, no? Now it's going to bind with the oxygen of this one. And then you have the hydrogen here. Normal as usual. See, everything else is going to be usually there like before. The only thing that's going to be removed is what? The hydrogen of this one and the OH of this one. And what is the product we have? Here, this one. Okay. So now you know this oxygen is going to be there. This is going to be the special part here. This part. Okay. Now, this is the special part that we have to think about. This bond here. Now, how do you call this bond? You call this bond as what? The glycosidic bond. Why? This is the bond that holds both the monosaccharides together. Now, do you understand? When the hydrogen of one monosaccharide and the OH of the another monosaccharide is going to be removed as what? Water. And that process is called what? There's a special name for that process, okay? You call that as condensation. When you remove water, you call it condensation. When you add water, you call it hydrolyzation. Okay, hydrolysis. Okay, so when you remove the water, that's called condensation, and that is when that bond is formed. So as a result, you get this disaccharide molecule. Okay, now this whole thing is a disaccharide molecule. Okay, do you understand that? Okay. Now this is how a disaccharide is formed. Now, what are the two terms I told you? We have, now we'll come across this. If you have a monosaccharide and another monosaccharide, they undergo what? Condensation. What is the meaning of condensation? Condensation means water is going to leave, water is going to be removed, and you form a disaccharide. Now this is one way of this happening. Now the other way is, you have a disaccharide molecule, okay? You undergo hydrolysis. Hydrolysis. What is the meaning of hydrolysis? When you add water, that is opposite of condensation. Then you result in monosaccharide and another monosaccharide. Do you understand the difference between these two terms? It's very important to understand, okay? Now similarly, you have in bio hydrophobic, hydrophilic. What is hydrophobic? Hydrophobic means you don't like water. Phobia means what? You're scared of water. Philic means you like, you love it. So hydrophilic, hydrophobic. Condensation, hydrolysis. These are opposite terms. You'll need that later, okay? For now, you don't need to know about that hydrophobic, hydrophilic, but later under plasma membrane and all, you have that, okay? Okay. So that's how a glycosidic bond is formed. Now you have to remember this, okay? Next, what do you have to remember about the disaccharides? Now we learnt about how the glycosidic bond is formed, no? Now don't remember it in paragraphs. Remember it like this. What do you have to remember? How that disaccharide is formed, what happens between them and all that. This rough idea is enough for you to write a whole essay. That's what I'm saying. Then you move on to how the examples that you have, okay? Now this was also there in your O level, so there is nothing much new, just for you to write that equation properly. So what are the three now? Three like very common monosaccharides that we know are what? Glucose, and then we have galactose, and then we have fructose. Okay, there's nothing to confuse in this. Three examples: glucose, galactose and fructose. Now glucose, when it binds with another glucose, now what are the combinations? Only a glucose molecule is going to bind with another glucose only. It can't bind with some other thing. Only a glucose and a glucose together. What are they going to undergo? Condensation. I'll just put C here, okay? C means condensation. And what are they going to form? maltose. Now when you write the equation, you put water as a product. Why do you put water as a product? Why? I told you here. When condensation happens, water and disaccharide are the results. Okay, water and disaccharides are the results of condensation. Here also, condensation, maltose and water. What does this mean? When you write the equation, if it is a condensation reaction, water is a product. If it is a hydrolysis, water is a reactant. You add water to the left side of the equation. Okay, and you make maltose. Now what is this maltose? Maltose is a disaccharide. Okay, then we move on to the next type of disaccharide, that is galactose. Now galactose is also going to react with what? Glucose. Glucose is like a common fellow. Okay, now glucose and galactose is going to react and they undergo condensation to form what? Lactose. Okay, lactose and water. Similarly, fructose and glucose react together, undergo condensation and form what? Sucrose and water. Okay, so what are the disaccharides that we have here? Maltose, lactose and sucrose. These are the three that we have. Now I'm just telling this. Now some of you all might find it very difficult to remember whether glucose is a monosaccharide or a disaccharide, something like that, right? So sometimes you might confuse of whether maltose, lactose and sucrose is a disaccharide or a monosaccharide. So how do you remember that? It's a very it's something like a dumb mnemonic that I made, a shortcut I made when I was doing my O level. So I just remember what are the three disaccharide examples. Masul. Okay, I know it's a very dumb example, but just remember it. Ma means what? Maltose. Su means what? Sucrose. L for lactose. These three are what? Disaccharides. Anything else is not going to be disaccharides according to me. Why? I don't have to know anything else beyond this. So masul is going to be what? Example for disaccharide. Okay. And then you have to know another very important information. I told you there's something called reducing sugar when we spoke about monosaccharide. Can you remember this? Reducing sugar. Now here, I told you all monosaccharides are reducing sugar. But when it comes to disaccharide, if there's a statement like all disaccharides are reducing sugar, that's wrong. Why? There are non-reducing sugars also. I told you don't have to know what is reducing and non-reducing. That is just out of syllabus. You just have to know whether it's reducing or not. So, we have one non-reducing sugar. What is it? Sucrose. Non-reducing. Now don't confuse whether this is reducing or non-reducing. Okay. So if this is non-reducing, the other two have to be what? Obviously reducing sugars. So remember this. All mono and disaccharides are sugars. All monosaccharides are sugars and reducing sugars. Disaccharides, except for sucrose, the other two are reducing sugars. But sucrose is also a sugar but not reducing. Okay, non-reducing. Now I know more than maltose and lactose, sucrose seems like a sugar. No, it sounds like a sugar. It's like more like, it has a sugary name, I would say. So just because sucrose sounds like a sugar, that doesn't mean that it's reducing. Remember, this is the exception. Sucrose is non-reducing. Maltose and lactose are reducing. So that's what you have to remember for disaccharides. Now see, disaccharides, what you have to remember, how a disaccharide is formed. When a monosaccharide binds with a monosaccharide, a disaccharide is formed. And what is the bond between the two monosaccharides that forms the disaccharide? Glycosidic bond. Now you have to know how that glycosidic bond is formed. How is it formed? The OH group of one monosaccharide binds with the hydrogen of the other monosaccharide. Only two monosaccharides are involved in this process and they undergo condensation. What is the meaning of condensation? Water is removed and also we result in a disaccharide. Now this disaccharide, here you can see how that bond is formed. Why? This water part is removed and hydrogen is going to now bind with oxygen. So this is called the glycosidic bond. Then you have to know the three examples, the proper equations. They can even ask in structured, in your part 2, you have two parts. Structure essay and essay. So in structured essay they can ask, give the equations for the formation of maltose, lactose and sucrose, or one of them or whatever. So here you can see glucose and glucose undergo condensation to give maltose and water. Similarly, you can see glucose is a common fellow for all these three. Okay. So what is different? Glucose, galactose and fructose. Remember, just like glucose, galactose and fructose are monosaccharides. Disaccharides are maltose, lactose and sucrose. I told you, if you find it very difficult to remember whether maltose, sucrose, lactose are monosaccharides or disaccharides, then what do? You remember this mnemonic, muscle. So muscle means maltose, sucrose and lactose. Now in that, sucrose is the exception, why? It's non-reducing. So here if you consider, now what are the sugars that you have in these equations? Glucose is a sugar, galactose, fructose, maltose, sucrose and lactose. Out of these, which are reducing sugars? Glucose, galactose, fructose, maltose and lactose. Why? Because all monosaccharides are reducing sugars and all disaccharides except for what? Sucrose are reducing sugars. So sucrose is the non-reducing sugar. So that's it you have to remember for disaccharides. Okay. Okay, so now under polysaccharides you have to know about the composition and everything. Okay, so first of all, there's a simple unique feature about polysaccharide. What is that feature? Firstly, they say that they are macromolecules and biopolymers. So these two terms are very important, macromolecules and biopolymers. Macromolecules and they are biopolymers. So after this we have another point, how are they made of, like how are they composed, right? So that's what we write in the right corner, right, usually. So under composition, this comes under composition. So you would have noticed in all our mind maps we wrote the composition in the right corner. So now that's like a permanent place to write about the composition. So composition, you can say they're made up of few hundred to few thousand monosaccharide subunits. Few hundred to few thousand. So like small few hundred to few thousand. So what are monosaccharides? Monosaccharides are the monomers. So few hundred to few thousand molecules are going to combine like this to form a total big molecule called the polysaccharide. Okay, so after this we move on to the nature. Now the nature, of course, we wrote in this side. This side we wrote for disaccharides also. So we'll write that here, or we can say features, no? Features. So for features you can write they are crystalline. What are the three features I told you and how to remember it? I told you to remember the normal sugar. Normal sugar is crystalline in shape, so here also it's crystalline. And then you add that normal sugar into water and you dissolve it. So how do you say? It's the solubility is what we described. Now in disaccharide I told you that they are water soluble. But can you remember I told you that polysaccharide is not considered as a sugar, although it has that saccharide term. Now here I wrote this shortly, but here this saccharide term will come, no? So polysaccharide, although it's considered as a carbohydrate, it's not a sugar. So if it's not a sugar, it's not soluble in water. Okay, so second feature we can say it's not considered as sugar. So you don't have to write the whole thing. Now this is a mind map. No, you don't have to write everything in sentences. Just write cross sugar, not considered as a sugar. And the third point is that it's water insoluble. Okay, so what are the three features? Crystalline, but it's not considered as a sugar, and it's water insoluble. And then you are going to speak about, now in disaccharides, what did we, in monosaccharides, how did we use the classification for monosaccharides, right? Then for disaccharides, of course, we didn't have any such special classification. We just mention about the ways they are formed. Here for polysaccharides, we are going to classify them according to certain criteria into two main categories. Okay, now they are in monosaccharide also two main categories. So you should be able to relate this. So here also you are going to bring them down into two main categories. So what are the two categories? The first category is according to their function. Okay, so based on their function, you are going to classify polysaccharides. So based on their function, what can you say? Some polysaccharides are going to act as storage polysaccharides, whereas some are going to act as structural polysaccharides. What is it? Storage polysaccharides. So what is the meaning of storage? They are going to store something. So some polysaccharides are going to be involved in storing some things, right? So we can say starch, glycogen, and also you can say inulin under this. Okay, so inulin, this inulin one, under the examples, in the very first time they introduce this to your book, that means like when you keep on reading your book, resource book, firstly they haven't mentioned this inulin, but later on under functions they have mentioned inulin also as a storage polysaccharide. So remember, we have three for now: starch, glycogen and inulin. And then we have another function. What is it? Structural function, which means they form the structure of something. So structural function, we can say structural polysaccharide. What are they called? Structural polysaccharide. Now structural polysaccharide, of course, you know, now cellulose is going to form the cell wall, right? So that's a very common function. These are not going to be involved in storing functions. Okay, and then you have hemicellulose, and then you also have pectin. Now what are these functions? These are going to be storage functions. And also in addition to this you have chitin. Same story like inulin, they haven't mentioned that in the first list, but later under functions they have mentioned chitin also as a structural. So remember, in your resource we have 3 storage polysaccharides and 4 structural polysaccharides. You can't pick and remember some, you have to remember all. So cellulose, hemicellulose, pectin and chitin. Now that's about the categorization based on their function. Now you have the categorization based on their architecture. Now before that, now here storage, no? So what do they store is important. Structural, where is it found is important. But I'm not going to write that now here, but maybe just to give an idea, I'll write it within brackets a very short way, okay? Because again we are going to come across this under functions of each polysaccharide. So starch, of course, they are going to store glucose in plants and chlorophytes. And so that's about the starch. Glycogen, of course, they are going to store glucose again as energy source in animals and fungi. Inulin, of course, they are going to store energy source in dahlia tubers. Okay. Structural, you know, cellulose and hemicellulose, both of them are found in the cell wall of plants and chlorophytes. Pectin, of course, it's found in the middle lamella of plant tissue. So these two, okay. Chitin is found in the cell wall of fungi and exoskeleton of arthropods. So that's what you have to know about the structure and function. Now once you know this example, you should be able to elaborate on how they are going to be a structure, how are they going to be acting as storage compounds. Okay. After this function, we have another criteria to categorize them. What is that criteria? That's called architecture. Based on the architecture also you can categorize polysaccharides. So what is architecture? How they are formed. So I told you now when it comes to disaccharides, some are linear and some form rings. Can you remember the examples for those? I told you that glucose and fructose, they're solid when they are linear and they become rings when they are in the liquid state. Now in the aqueous media. But now that's not the architecture we are speaking about here. Here we are speaking about whether they are linear or branched. So if it's linear, there's no connection like this here. Like a bunch of strands is linear. If they have bridges between those strands, like connections between those strands, then you call it what? Branched form. So if it's normal way, you call it linear, and if it's branched, you call it when there are like cross links or whatever, you call it branched form. Okay, so based on that, you can classify them as linear and branched when it comes to polysaccharide. So what are the linear polysaccharides? Now when you see the name itself, linear ones have a very simpler version of that same name, but the branched versions have a more complex name, like you can sense it. Now for example, I'll tell you cellulose. Cellulose isn't that simple, it sounds very simple, no? But hemicellulose sounds a bit complex. Why? Like a few cellulose are going to be like stacked together and they are going to have cross links, something like that. Do you sense that feeling? Hemi. Okay, so same thing, amylose comes as linear, whereas amylopectin comes as branched. Now you can see amylopectin, it sounds very complex, so this is not how you really remember it. But once you see hemicellulose, amylopectin, they sound more complex, so you can remember those are branched forms. And then you also have glycogen for branched form. Now glycogen also sounds somewhat heavy, right? These are just like, you know, a gut feeling, like, you know, they sound branched. That's what I'm saying. Okay, so now remember this. Now there are some examples that are commonly found in both. So now based on their function, you categorize them as structure and structural. Then based on their architecture, you categorize them as linear and branched. So there can be overlaps. Now for example, now they are not going to ask you very simple ones. They are going to ask you, give an example of a linear structural polysaccharide. Then you have to see what are the common ones. Linear and structural cellulose. Okay, and then I can ask you, give me an example of a branched structural polysaccharide. Then that's hemicellulose. Branched storage polysaccharide, then that's glycogen. So you should be able to link it in that way also, not just the direct way of how you remember it. Okay, so that's like application based questions. Okay, so now after we are done with this, so we'll quickly wrap through what we did for polysaccharides. Polysaccharides, I told you, now always under each category, the top part we remember something unique about that particular group. So here it's macromolecules and biopolymers. Then under the composition, we remember it's made up of few hundred to few thousand monosaccharide subunits. And then under features, I told you to remember that sugar scenario. Here of course it's not a sugar, it's water insoluble, and but it's crystalline. Okay, so don't think this is not crystalline. Now that's another this is also non-crystalline. Sorry, there's a small mistake. This is also non-crystalline. Okay, so what can you remember? Polysaccharides have features which are completely opposite to that of disaccharides. So they are non-crystalline, they are not considered as sugars, and they are water insoluble. Just correct this part. This is not crystalline, non-crystalline. Okay, non-crystalline. And then you are going to categorize polysaccharides based on their criteria. So we have function and architecture. Based on their function, you are going to have storage and structural polysaccharides. So storage means they are going to store something. So starch, glycogen and inulin. Structural means they are going to form a structure of something. So cellulose, hemicellulose, pectin and chitin. Then based on the architecture, you have linear and branched. So linear means they are going to be found in strands. So cellulose and amylose. If there is going to be like cross links between the strands, you call it as branched. Example, hemicellulose, amylopectin and glycogen. So that's it for the polysaccharides that we have. Now you have to have an idea about the overall picture. Why? Now we are going to speak about the functions and a bit more depth in terms of what they do. Right, so I told you, now like we'll go back to the previous explanation that we had. Firstly, what did we say? Carbohydrates are there. And I told you carbohydrates are going to be divided again into three main types. What are the three main types? Monosaccharides, disaccharides, polysaccharides. Now you should be very familiar with these terms now. So I'll just write mono, di, poly. Now under monosaccharide, I told you, monosaccharides, you are going to give examples for their functions. So there are three functions mainly. Firstly, they are going to act as energy source. Now what is the common monosaccharide that you remember? Very common ones. Now I'll give you a hint. This is something we directly consume when it comes to sports meats and stuff. So what's that monosaccharide? That is glucose, right? So just remember along with that glucose example itself, monosaccharides are going to act as an energy source. They are going to act as an energy source. Secondly, you can remember they are going to be making the building blocks of disaccharides and polysaccharides. So they are going to make what? Di and poly. Who are the people who make the di and polysaccharides? It is monosaccharides. Okay, so they are going to be building blocks, and you can give examples of disaccharides and monosaccharides that they form. Then they are also going to be building blocks, or we can say components of nucleotides, like we can say DNA, RNA. For now, remember this. When we come to nucleic acids, you will understand. Okay, so monosaccharides, of course, we have three functions. What are they? They are going to act as an energy source. They are going to form disaccharides and polysaccharides. They are going to be components of DNA and RNA. Now we move on to the disaccharides. Disaccharides, I told you a mnemonic to remember. What is it? Muscle. In case you always struggle to remember the example. Ma for maltose, Su for sucrose, L for lactose. Now under the under the functions in your book, they haven't given anything about maltose. Okay, but for sucrose, of course, they have given two functions. First one, translocation in phloem. Now what is phloem? You would have heard this term even in O levels. Now in the plant, now you make sugar, right? Sucrose is made. Now you're going to transport that sucrose throughout the plant. You're going to transport it, then only the plant will get energy to do the function that it does. So in a plant, if you're going to transfer that sucrose, that has to be taken place through a special type of a tube. Okay, a special type of tube that is going to extend throughout the plant to the leaves and everything. So this tube is called phloem. So the process of transporting that energy source through the plant is going to be called phloem translocation. So phloem translocation, in that who is involved? Sucrose. Okay, then you have, now even phloem translocation will come as a separate whole topic later under your fourth unit. Okay, and then it's also very similar to the first point. They are going to act as a storage sugar in sugarcane. Now in sugarcane also, you know, it's very sweet, right? So in sugarcane, what is found as a storage sugar? So don't write everything, just write sugarcane. It's going to be found as a storage sugar in sugarcane. What is it? Disaccharide. Okay, and then you have about the polysaccharides, but before that we have, I told you, sucrose we are done. Lactose. Lactose, what do we have about lactose? Lactose, of course, they have said they are storage sugar in milk. In milk, you find, just draw a cup of milk and you say storage sugar in milk is going to be what? Lactose. Okay, so three things about disaccharides. Sucrose, you have two examples, phloem translocation, then second one, storage sugar in sugarcane. Lactose, you have storage sugar in milk. Then we move on to polysaccharides. Now polysaccharides, of course, I told you there are so many examples. So it's better to go with the previous categorization I told you. Here this one. Now here we have all the polysaccharides that we need for our work. Okay. So first of all, we'll start with starch. Starch is going to be found where? What are they going to store? Glucose. So they are going to store glucose. I'll write G here. Okay. They are going to store glucose in plants and chlorophytes. Then glycogen is also going to store glucose in plants and chlorophytes. Inulin is going to store Chlorophytes means green algae and all those. Okay, so green plants and green algae, all those you're gonna, not only in green plants, plants and green algae and all those, you're gonna find starch and glycogen. Starch in glycogen, of course, it's found in animals and fungi. Okay, and then you have inulin. Inulin is going to be found in the tubers of dahlia. Now dahlia flower has like in those lotus flowers, you have like tubes inside it. Okay, tubers you call it. So inside that tube you're going to find inulin, and that inulin is going to be the one that stores fructose as an energy source. Okay, so as an energy source, this is the one that's going to store fructose. And then under structural, you can remember cellulose again in the cell wall of plants and chlorophytes. Hemicellulose, it's going to be found in the cell wall of plants. Pectin is going to be found in middle lamella of plant tissue. Middle lamella means, now you know you have a cell. Okay, here also you have a cell. Now that the joining part of these two, that's going to be called the middle lamella. Like, you know, now there is a cell like this. The sticky polysaccharide between the cell wall is called middle lamella. It's like a sticky thing. Now there are cells like this. No, the one that's found between this, that's going to be called what? Middle lamella. This is called middle lamella, the one in blue. So this sticky polysaccharide is pectin, and that's going to be forming the structure of the middle lamella of plant tissues. Then chitin. Chitin is very important. That's going to be found in the cell wall of fungi. You know fungi, no? For example, mushrooms. Now, in the cell wall of that, you're going to find chitin. And also in the exoskeleton of arthropods. Now, you know, in a cockroach, okay, I'll just draw a cockroach. You have a shell kind of a thing on top, no? Now that shell is going to be called the exoskeleton. In that exoskeleton, you're going to find chitin. So what are the polysaccharides that are found in animals? If I ask you, you're going to say glycogen. Why? Glycogen is found in animals and fungi. Then you have to say chitin. Chitin is found in the exoskeleton of arthropods. Okay, you don't consider fungi as animals. I'm speaking about direct animals. Then if I ask you what is the type of polysaccharides that are found in plants, then starch is going to come, inulin is going to come, cellulose, semicellulose, pectin, you can say. Okay, so that's it about the polysaccharides. So remember, there are key terms to mention when you're speaking about the structure. Firstly, we are speaking about the storage. You have to say stores. This is a format of the sentence you have to write. Stores, what does it store? Stores dash in dash. Now, for example, if I take starch, stores glucose in plants and chlorophyte. Glycogen stores glucose in animal and fungi. Inulin stores fructose in tubers of dahlia. Do you get it? Now, when it comes to structural, there's a format of sentence you have to always write. It's found in dash of what? Now, cellulose in the cell wall of plants and chlorophytes, and then pectin, or we can say hemicellulose, in the cell wall of plants. Then pectin in the middle lamella of plant tissues. Where is it found? Okay, if you put it in this format, you can score all the marks. Then pectin, okay, chitin in the cell wall of fungi and in exoskeleton of arthropods. Do you get the format? Okay, so now we are done with the polysaccharide also. So that's it for carbohydrates. Now, if you break this complex topic, now you know carbohydrate seems like a very big topic, but if you are going to normally study it, it's going to be seeming like a big chunk. But you broke it down. How did we first break it down? Firstly, we broke it down into 3 types: mono, di and poly. Then we broke down each mono also into different chunks, di also into different chunks, poly also into different chunks. Now you have small pieces, but you have the connection between each piece. Now that's the whole point of this mind map. Why? You can relate into different parts of the mind map, and you're going to like understand it more analytically, and it's easier to revise also. Why? Before the exam, you don't have to read your resource. You just have to look at this mind map and see All these points are there. There's nothing more than this that can be questioned in the exam. It's just about you using these key terms and making a whole sentence and writing it there. Okay, so that's the whole point of using a mind map to study bio.